Hey there, my name is Kyle Proctor. I'm an undergrad student here at the University of San Diego, majoring in environmental studies. My research project looks at the delayed settling behavior of marine snow aggregates as they pass through sharp density gradients. In particular, we wanted to see how the growth phase of phytoplankton cultures used to form these marine snow aggregates would impact their settling. Marine snow aggregates are small particles found in the open ocean composed of microorganisms, sediment, and detritus. Previous studies have shown that the settling velocity of marine snow aggregates decreases significantly at sharp density gradients in a phenomenon referred to as delayed settling. Additionally, other studies have shown that phytoplankton cultures actually have different physiological attributes in different growth phases. With these factors in mind, the objective of this study was to determine how the growth phase of phytoplankton cultures used to form marine snow aggregates would affect their delayed settling velocity at sharp density gradients. To go about doing this, we grew a culture of the diatom Thalassiocyra westphalogii in 4 separate flasks, and we determined 4 distinct growth phases: the early exponential, the late exponential, the early stationary, and the late stationary. When the culture reached one of these predetermined growth phases, we would remove the flask, dilute it to roughly 25,000 cells per milliliter, and place that dilution into a roller tank on a roller table. It would roll on that table for about 3 days in order to induce the formation of marine snow aggregates. Once we had these aggregates, they would be removed by a pipette and transferred over to a 2-layer tank, which can be seen here in figure 1. The top layer of the tank has water of the same density that the aggregates were formed in, and the bottom layer of the tank has water of a slightly higher density. In the middle, there's a sharp density transition which we were able to form using a diffuser. Once these aggregates are settling, we record their settling path using video equipment. That can be seen here in figure 3. On the y-axis, we have the height of the tank, and along the top, you can see the time in red. For the first roughly 40 seconds, the aggregate settles kind of as you would expect. It falls relatively linearly until it reaches this yellow line, which represents the sharp density transition. It remains at that sharp density transition for roughly 15 minutes before it continues to settle again linearly in a few seconds. You may wonder, why would that happen? This is due to a phenomenon referred to as diffusion limited retention. Because of the extremely porous nature of these aggregates, it takes time for that lighter water to be diffused outward by the more dense water in the lower portion of the tank before the aggregate can continue settling. Here in figure 4, you can see 2 figures that come from our MATLAB analysis of this data. Figure A represents a position graph. We have the vertical location on the y-axis, and this roughly parallels what we see in figure 3. It drops sharply, stays at the density transition, and then continues dropping. In figure B, we have the velocity of the aggregate graphed on the y-axis. Using this information, we were able to find the delayed settling time, or DST, for each aggregate. The DST is defined as the portion of time where the aggregate is moving at 90% less than the maximum velocity in the lower portion of the tank. The DST here is represented by this green line. By collecting the DST for each of the aggregates in each of the growth phases, we were able to create figure 5 seen here. When comparing the mean DST for each of the growth phases are represented in the various colors along the x-axis, and DST is represented on the y-axis in minutes. We found that the delayed settling time actually increased as we moved into the later growth phases. This means that these aggregates were taking longer to settle as they progressed in their natural growth. Surprisingly, in the final growth stage, the late stationary, it actually took the delayed settling time decreased, so it didn't take as long for these aggregates to settle outward. Additionally, there was a significant difference between each of the neighboring growth phases. This gives credence to the idea that there is some sort of significant physiological change happening between the growth phases, which may impact delayed settling time. Now, this information has many implications for the biological pump as a whole and carbon flux. I think it is important that we do further research to understand how anthropogenic effects may impact the growth phase of phytoplankton in the open ocean, which will then lead to changes in the delayed settling time of these aggregates and ultimately a change in carbon export. It's also crucial to understand that roughly 1/3 of all carbon that is sequestered in the natural system is sequestered by the ocean. So until we have a better understanding of how this process is occurring and at what rates, it will be very difficult for us to move forward in the best possible way. Thank you.